So, hello everybody.
Hello.
My name is Katrine Johannesen, and I'm a medical doctor and a PhD student at the Danish Epilepsy Centre. Very happy to be here today. At the Danish Epilepsy Centre, we see all the patients in Denmark who have epilepsy, and we do almost all of the genetic testing on these patients in Denmark. So it's quite unique. Today's topic is, as you see, SCN2A and what's new. There will be less talk about cars and more talk about the clinic compared to Dennis' fine talk on SCN2A earlier this year. So for a general introduction to genetics and functional consequences of mutations, I recommend that you watch that talk. My background for talking about this is that I was one of the shared first authors on the brain paper on SCN2A that came out in the beginning of this year. And this talk is based on that paper. So yeah. So let's get started. This is just the basics, a brief introduction, so we're all on the same page. SCN2A is one of the more than 20,000 genes in the human genome, and it encodes this voltage-gated sodium channel that is very widespread in the human central nervous system. These channels are located at the axon initial segments and nodes of Ranvier during early development, so at the synapse. And well-functioning channels are essential for the conduction of action potentials and thus for maintaining this fine balance between exciting signals and inhibitory signals in the brain, and for the communication between neurons. So these channels are very critical for normal early neuronal development, which you can see in the diseases that they cause when they are malfunctioning. So in this study that we did, we looked at exactly what happens when you have a dysfunctioning channel. So we collected a total of 201 cases, both cases that had been previously published as well as new cases. And what's depicted here is the distribution of phenotypes. And as you can see, the vast majority, the green, red, and yellow, that's 69%, have some kind of developmental and epileptic encephalopathy. And encephalopathies are diseases in which the patients suffer from developmental delay and where the epileptic activity in the brain causes further damage to the cognitive development. So these patients are seriously ill and will often require help in their everyday life. We also found two minor subgroups, the blue and the yellow, and this is the benign familial epilepsies and the intellectual disability and or autism without epilepsy. I will talk a little bit about these later. So overall, we have these four main groups who all have disease due to variations in SCN2A. And these four groups are benign familial epilepsy. It's epileptic encephalopathy with early infantile onset. It's epileptic encephalopathy with infantile childhood onset. And it's intellectual disability and or autism without epilepsy. And as I mentioned in the beginning, here in Denmark, we have very good data on childbirth, and we have a very specialized epilepsy hospital. So therefore, we were able to calculate the incidence to 1 in 87,608 births. So a very rare condition, But in regards to the monogenic epilepsies, SCN2A is probably one of the most common causes, next to, for instance, SCN1A, which causes Dravet syndrome. So I will try to present the clinical characteristics in each of these 4 groups. The benign epilepsy subgroup, although these variants and epilepsies usually run in families, we found de novo variants in 6 probands. But as with all things, it has to start with somebody. So this is probably just where the disease starts. Common in these patients is quite an early seizure debut. So 50% of them present with seizures within the first month of life. Their seizure types are focal clonic, tonic, and tonic-clonic, and often the seizures will appear in clusters. They all become seizure-free, hence the word benign, and they all have normal cognition and behavior. Interestingly, within like the last 2 years or something, as we've seen a subgroup of these benign epilepsies, and these are children who have either a variant in the alanine 263 valine or in the arginine 1882 glycine. And they have an epilepsy that's characterized by a little bit longer and more severe seizures than what we would expect from these benign epilepsies. And they sometimes suffer from status episodes, but they still become seizure-free within like the first year of life. Then during their second year of life, they develop episodic ataxia. And I have a small video that I hope we can see of a child with this an ataxic episode. It's one of the Danish patients, so the mother is talking Danish. And I hope that you can all see that she has these, how it gets very prominent. That she has this ataxic episode, and it's of discomfort to her. And as of yet, we do not know why these children develop episodic ataxia, and so far we've found no good treatment for these episodes. This girl, this was like 2 years ago, and she still suffers from these episodes. And it seems that sodium channel blockers do not have a good effect. But we do have some colleagues in Germany that are working more with this episodic ataxia. Yeah. So moving on, the second group is the patients with the encephalopathy with early infantile epilepsy. They, per definition, have intellectual disability and epilepsy onset within the first 3 months of life. Within this group, it's included Ohtahara syndrome and epilepsy in infancy with migrating focal seizures. These patients have focal, tonic, tonic-clonic seizures or epileptic spasms. Their EEG often show a suppression burst pattern or multifocal spikes. About 2/3 of them suffer from severe intellectual disability, and most of them have other difficulties than epilepsy, and these include like autism, hypotonia, microcephaly, and movement disorders. And when we did the study, we found that 7 patients were deceased, and that they died from severe infections or status epilepticus. Then we have the third group, which is the encephalopathy with infantile childhood onset. who also per definition have intellectual disability, and then they have onset of epilepsy after 3 months of life. And this group includes West syndrome, Lennox-Gastaut syndrome, myoclonic atonic epilepsy, and Dravet syndrome. Seizure types are mainly generalized tonic-clonic seizures, absences, and myoclonic seizures. And their EEGs show general spike and wave or multifocal spikes. We also found 4 patients in our study who developed ESIS, that's electrical status epilepticus during sleep. About 1/3 have severe ID, and comorbidities are also prevalent in this group. And includes autism, hypotonia, and movement disorders.
Can you hear me? Is it possible to ask a question now?
Yeah.
So, I know Dennis and others have sequenced such patients and mapped where the mutations lie. Mm-hmm. Is there any rationale in terms of the channel itself and the mutations that accounts for these different categories, these different phenotypic categories? So
you mean as to where in the gene the mutation is? Yeah. I will get back to this, but no, we have not found like a link between where the variant is and the phenotype.
But Dennis knows that some 3-dimensional mapping of the mutations onto models of the protein have been done with a colleague of his at the Stanley Center, and I wonder whether that methodology has been applied to your groups of patients with the...
So actually we are working closely with Katrine and her mentor Ulrike, and this is the data sets we are actually using at the moment. These are the variants we are looking at, and I think we have some preliminary results which we have not yet communicated with Katrine, but we are working on this, and we are working also on the paper.
Yeah, and so the other question is this a mixture of gain of function mutations and loss of function mutations, or is it all in one direction?
I think Katrine will talk about this in the next slides.
I will get back to that in a couple of slides. Yeah.
All right, thank you very much.
Hi. Can I ask one question?
Yeah.
Can you describe the movement disorders at the bottom of your slide? Is that in more detail?
Yeah, unfortunately, this is a retrospective study. So for a lot of the patients, we only know that they have a movement disorder and not like the specifics of it.
Okay, because I met a patient through the SCN2A Foundation that had cerebral palsy, and I wasn't sure if you came across any patients with cerebral palsy.
Yeah, some of them definitely have that.
Thank you.
Yeah. Okay, so let's just take the last group. That's the these patients that do not have epilepsy, and this. In our study, this was quite a small group, but most likely this is because they are underdiagnosed, because maybe they haven't been genetically tested, because genetic testing is not that prevalent in this category. So right now there are several efforts going on, and it does seem that SCN2A might be one of the bigger monogenic causes of autism spectrum disorders, for instance. So what we found from these few patients is that they had moderate to severe intellectual disability, autism, or a few of them schizophrenia. But as I said, data was quite sparse, especially in these patients. For the 3 groups in which the patients do suffer from epilepsy, we tried to look at treatment response. Again, this was a retrospective study, but we found that seizure freedom was achieved in, of course, 100% of the benign group, and in 57% of those with epilepsy onset before 3 months, and in 34% with onset after 3 months. We also tried to see if the fact that they got a genetic diagnosis made a difference in treatment strategy. As this was a retrospective study, most of these patients got their genetic diagnosis relatively late in the disease course, but we did see some in the early infantile group who changed their treatment to sodium channel blockers and had an apparent good effect of this. So we tried to dig a little bit more into this and evaluate the treatment response in these two encephalopathy groups. And this is a picture showing those two groups. And down, like in the middle, you can see the anti-epileptic drugs. And those in colors are sodium channel blockers. So sodium channel blockers are thought to work well in SCN2A patients. The hypothesis is that the channel is hyperfunctioning. Thus, the variance, as we discussed a little bit earlier, has a gain of function effect. And by adding a sodium channel blocker, then you would hope to diminish this hyperfunctioning. And it does seem to work well, but as you can see, it's only in the group with onset before age of 3 months. Where there is a significantly better response to sodium channel blockers. We did not find this effect in the group with onset after 3 months, who, as you can see on the bottom right, responded better to non-sodium channel blockers. So we think this is quite interesting, and that maybe it could have something to do with the expression of SCN2A, which is highest in the neonatal period, or could have something to do with the actual function of the mutation in SCN2A. I just want to mention that on the top here you see phenytoin, and I just have to mention that these are, to have a real good effect of phenytoin, you need quite high doses in these patients. And they tolerate that very well, actually. So some of these differences in the treatment response can be found in the genetics. We found that the benign group and those with onset before 3 months of age all had missense variants in SCN2A and missense variants only. Whereas the encephalopathy group with onset after 3 months and the intellectual disability autism group had both missense variants as well as nonsense variants, who are per definition loss of function variants. I also want to mention that we found several recurrent mutations. And those I mentioned here are some of the extremely recurrent hotspot mutations, what you want to call them. The A263 variant that I mentioned earlier with benign epilepsy, and then the episodic ataxia. But we did also see it in a couple of patients with encephalopathy. Then there's the 853 variant that causes a severe encephalopathy. The majority have West syndrome. The 1319 position seems somewhat milder variant that causes either benign epilepsy or encephalopathy with seizure freedom. And at position 1882, it depends on the amino acid substitution. So those who have a glycine substitution have a benign epilepsy, and whereas all other substitutions, they will develop a severe encephalopathy. And then we did functional testing on 4 variants. The first two, the 1597 and the 1423, were found in patients with severe encephalopathy, but one of them showing a good response to sodium channel blockers. And we did see that these 2 variants were gain of function, whereas the 899 and 1622 variants were found in patients also with severe encephalopathy, but with adverse reactions, so worsening of seizures to sodium channel blockers. And we found that these were loss of function. So this goes quite well with what I talked about before, that there is a preferential response to sodium channel blockers in the variants we suspect to be gain of function, and no response or perhaps worsening in those we think are loss of function.
Why, if you have loss of function, do you get epilepsy?
Sorry, say that again.
Why, if you have loss of function, do you get epilepsy?
Yeah, that's a really good question. I don't know the answer to this, because we see the loss of function also in the group without epilepsy, right? So we don't know why this causes epilepsy as well. And maybe it's something about, I don't know, maybe it's something about the expression of the SCN2A in the neonatal period or something like that, but we simply do not know yet.
Yeah, from what we discussed this also here. So we don't know this, it's pure speculation, but it could be that the gain of function phenotype would be more like an imbalance disorder, where you have a channel which is functioning too hard and you can block that. Whereas the loss of function is more like a developmental circuit disorder, yeah, which is also would support that because you cannot treat it, with blockers. But who knows? But it's an important question also in terms of hopes for drug or the drug development and timing of drug development, or timing of when you give the drug.
Are people in the field creating mouse models or cellular models with representative mutations of the different phenotypes that you're talking about, so something mechanistic can be understood about this, or that is not yet underway?
Not us in Denmark, but some of our German colleagues are doing functional studies in several other variants to try to shed a little bit more light into this dilemma.
So as far as I understood, I think there are like 3 to 4 mouse models out there which people study. Most of them are missense mouse models, which are gain of function epilepsy, early onset epilepsy ones. And there's one mouse, I think, where people look at conditional knockout of the allele, and then there's also the head knockout mouse. But I don't know if people look at this systematically by phenotype. So obviously, people looking at the loss of the full allele, full gene, and gain of function variants. But I don't know if they look at subtypes specifically yet at this point.
No.
Yeah.
I have a question. This is Leah. Katrine, on the last slide, the one that shows the recurrent variant.
Yeah.
I wanted just to make a comment. The R853Q, in our group, we see a lot of movement disorders associated with that variant. I just wanted to share that.
Yeah, it is one of the most severe recurrent variants. It is. Yeah.
And it's always the same movement disorder. I can't remember the full name, but we call it chorea. It's the dancing-like movement disorder. Thank you.
Yeah, they are quite similar, these 853 patients, actually.
Okay, thank you.
Okay. So I just want to conclude that SCN2A is one of the most common monogenic causes of epilepsy, and that these patients can be distributed in these roughly 4 groups: the benign epilepsies with a good outcome, but with the small subgroup who have episodic ataxia. Then the encephalopathy group with onset before 3 months with missense variants that are gain of function and usually show a good response to sodium channel blockers, and the encephalopathy with onset after 3 months, where we see both the missense and the nonsense variants, where we think that the missense variants are likely to be loss of functions. and thus have poor or adverse response to sodium channel blockers. And the final but probably underestimated group are the patients who do not have epilepsy but suffer from either intellectual disability or autism. So what's next? We are still collecting SCN2A cases. So if you are out there and have a patient with SCN2A, please do not hesitate to contact me, and I'll be happy to include him or her in our research. Also, we really want to try to set up a clinical prospective trial investigating the treatment response in these patients. And this will have to be in collaboration with probably some of our European colleagues. Then there is the research in autism that I mentioned. This is mainly led by Stephen Sanders, and I think that, yeah, we probably will see some of these results quite soon. And yeah, last we would like to do like more functional studies on other variants to help us understand this, like the pathogenesis of these mutations. So with that, I just want to say thank you, and these are all the people that contributed to the brain paper. Yeah, and I'll take questions if there are any further questions.